Online now. Good morning, ladies and gentlemen. And good morning means all over the world a different time. I know that Rebecca is coming from Brazil, I think. Is it true? It is madrugada now. So a lot of different times all around the world. And we are trying to speak already this morning about biotic stresses. This is the poster presentation session and the ongoing presentations of the oral contributions we had yesterday. I would like to ask Manuel to show us the first video from Tabea Allen, and afterwards we have time for some questions from you, from the audience, and of course from you as the contributors as well. From the audience should ask questions via the session Q&A. There we can follow your questions and then respond to them. Tabea L. or Manuel, show us the first presentation.
Co-authors, I would like to welcome you to our poster presentation on Iphossoma lafigmae, a solution for classical biocontrol of the fall armyworm Spodoptera frugiperda. The fall armyworm is one of the most damaging pests in its native environment in tropical and subtropical America. It arrived in Africa in 2016 and has since spread throughout the continent onto Asia and Australia. There it is causing huge environmental, economic and social damage, threatening the food security of 200 million farmers in Africa alone. Farmers respond by using pesticides, amongst which are also highly hazardous pesticides. Biological control might be a more sustainable solution. The egg parasitoid Telenomus remus has recently been recommended for augmentative biocontrol, so for the mass rearing and mass release of it in Africa. Classical biocontrol, so the introduction of a natural enemy of the fall armyworm from the Americas into Africa, should focus on a larval parasitoid, so a parasitoid which attacks the larval and not the egg stage. Iphossoma lafigmae is such a larval parasitoid and a potential classical biological control agent. The objective was to assess what knowledge on the biology of the parasitoid is already existing, to identify its natural distribution, to have an overview of the parasitism rate it achieves in the field, and to determine what other parasitoid co-occur with it. We chose to approach this by systematically searching for literature and use two keyword searches. For the first one, we used the keyword Iphossoma in Web of Science, Agricola, Cap Abstract, and Food Science and Technology Abstract. The second search we did in full-text Google Scholar. This resulted in 121 initial hits, of which we retained 44 papers in English, Portuguese, and Spanish. We saw that Iphossoma lafigmae has a very wide distribution, reaching from the south of Brazil until northeast Mexico. In regards to the parasitism rates, the study has certain limitations. On the one hand, only sites where Iphossoma lafigmae occurred were selected. This leads to an overestimation of parasitism rates. On the other hand, sampling techniques tend to underestimate parasitism rates. Samples were mostly taken from standing plants with a tendency of sampling larvae which are too old for Iphossoma lafigmae. Also, the parasitoid does not form cocoons easily in rearing, thus may not have been recorded. However, the results show that in literature it has mainly been recorded as attacking the fall armyworm and not any other species. This means it is highly specific. However, there are two exceptions. It has once been recorded as a parasitoid of a specimen of Alabama grilacea and also of Anticarsia gematalis. With the latter, it is unclear if there was just one specimen found or multiple ones. On average, 25% of all fall armyworm larvae were parasitized. Mean parasitism rates by Iphossoma lafigmae was at 4.8%, however, varying much between different locations from 0.7% in Sao Paulo, Brazil to 14.5% in Sete Lagoas, Brazil. In regards to other parasitoids occurring together with Iphossoma lafigmae, Chilonus insularis is the predominant parasitoid, followed by Iphossoma lafigmae. Cortes Madrigal observed a tendency that Iphossoma lafigmae predominated in weedy diverse fields, whereas Chilonus tended to predominate in less weedy fields. To the conclusions. The natural distribution is restricted to the neotropics. This makes it a potential candidate for most of fall armyworm in invaded Africa. However, climate matching would be necessary to get a better idea of its potential distribution. It is also a predominant parasitoid in vegetatively diverse system. Agricultural systems in Africa are more diverse than in the Americas. This is in favor of a successful establishment of the parasitoid in Africa. It is also highly specific to the fall armyworm in its native environment. However, to assess its suitability as a classical biocontrol agent, host range testings would need to be conducted. Thank you for your attention, and I'm looking forward to your questions.
Thank you very much, Tabea. Did you already publish this review anywhere?
No, it's not published yet. We're planning on publishing it in the next month.
So send it to the Journal of Plant Disease and Protection.
I will note this down then in this case.
I'm the editor there, and this would be very interesting for us.
Okay. Oh, then I'm very excited because we've been looking for a journal.
Yes.
Yeah.
The question which arises is the question of specificity, like you did. No? So to liberate the organism in Africa is difficult to decide. What is your opinion to this in more detail?
Yes, there needs to be more testing, that's clear, because at the moment you cannot book. That would be unsafe to just release it like that. It looks very good from literature point of view, but yeah, we need to do more specificity testing. Currently, it's being done at CAPI in Switzerland, so we're working on it at the moment, but the first results will be published also in a couple of months, so then we will know more. But it looks quite good for the moment. Yeah.
Thank you. Yeah. Are there more questions? I can't see questions from the auditory. I hope we have auditory. I don't know this. Are there questions from you within the Zoom session? That's not the case. So we step forward due to the fact that we have only very few time. And who will be the next? The next will be the presentation of Itorobong. And Manuel, you can play it, please.
Good morning, the chairman of this session and fellow researchers. I'm presenting a paper titled Response of Solanum lycopersicum to Tuta absoluta and Glomus clarum using simple sequence repeat markers on behalf of myself, Nsi Itorobong Mabel, and Dr. Olaoyi Odunayo Joseph. Introduction. Tomato belongs to the family Solanaceae and order Solanaceae. It is sensitive to a number of diseases and often attacked by pests, of which Tuta absoluta is one of them. Over the years, several approaches have been employed in the management of Tuta absoluta, but there is limited information on the use of the environmentally friendly arbuscular mycorrhizal fungi, Glomus clarum, and the identification of molecular markers that is associated with the resistance of tomato to Tuta absoluta. This research was aimed at investigating the response of tomato to Tuta absoluta and Glomus clarum using morphological and molecular techniques. Materials and methods. 8 tomato varieties were used in this study, and complete randomized design with 3 replicates and three treatments, which comprised of Glomus clarum and Tuta absoluta alone, and control, which was uninoculated. Results. Table 1 shows growth, agronomic, and yield characters of tomato as affected by Glomus clarum and Tuta absoluta. From this table, we see that Glomus clarum and Tuta absoluta treated plant was significantly different across all characters from Tuta absoluta alone plants and control plants. Table 2 shows gene diversity, allele number, and polymorphic information content of the SSR primers that were used for this study. From here, we see that primer LEAAT005 had the highest allele number of 4, had the highest gene diversity of 0.73, and had the highest polymorphic information content of 0.67, while primer LEGAA003 had the highest major allele frequency of 0.67. Figure 1 shows effects of treatment on percentage incidence of tuta absoluta at different growth stages. From here, we see that Glomus clarum treated plants, which is signified by the blue line, had reduced percentage incidence of infestation of tuta absoluta, while figure 2 shows effects of treatment on percentage severity of tomato infested with tuta absoluta at different growth stages. From here, we see that Glomus clarum treated plants, also signified by the blue line here, had reduced percentage severity of infestation of tuta absoluta. Discussion. The reduction in percentage incidence and percentage severity of tuta absoluta infestation, and also the significant difference that was observed between Glomus clarum treated plants and other treated plants could be as a result of Glomus clarum induced optimum nutrient uptake, damage compensation, and activation of tomato defense mechanism, which was also reported by Olaoye et al. 2013, Olaoye et al. 2014, and Olowe et al. 2018. Conclusion and recommendation. Plants treated with Glomus clarum reduce the infestation of tuta absoluta. Also, Prima LEAAT005 and Prima LEGA003 detected the highest genetic polymorphism and could be used in marker-assisted breeding of tomato and other Solanum species. Thank you for listening.
What a cool presentation, Ito Hobong. This was the first presentation I saw on this Tropentag in this way. So congratulations for this way of presentation. My question is really everyone knowing what is Glomus clarum? Glomus clarum is a mycorrhizal fungi, a fungus which is colonizing the roots. And you are speaking about the interaction with an insect feeding on fruits, on leaves, and so on. So that's the question. Which mode of action might this mycorrhizal fungus in the root have to tuta absoluta in the leaves? How does this work?
Thank you very much, sir, for your question. First of all, the mycorrhizal fungi colonizing the root helps the plants to gain more nutrients and then it provides a buffering effect, thus making it able to overcome the whatever pressures that are coming from the insect. Also, apart from the nutrients, it helps in damage compensation, such that if the insect is infesting on the plant is more able to overcome the effect of the insect on it.
Mm-hmm.
Thank you.
Yes, that's one possibility, that it is an induced resistance or induced tolerance of the plant. But more and more we learn from other examples that there are changes induced by mycorrhizal fungi of the gases coming out from the leaves. The odor of the plant is changed and the insects are irritated. So this is a very interesting example, and I will keep it in mind to come back to you to a later time. Thank you very much. I still do not have questions.
Okay, sir. Thank you very much.
I still do not have questions from the auditory. Are there further questions from our Corona here? Yeah, Tanya?
Do you hear me now?
Yes, I can hear you.
So you tested Cultivars, did I get this right?
Ma, I can't hear you.
Can I try with chat?
We can't hear you. Sorry, Tanya. Further questions? So if not, we would step forward, Manuel, with the next presentation.
Hello, my name is Nara, and I will present the poster The Use of Agrochemicals and Mortality by Stomach Cancer in Brazil between 1979 and 2015, developed in the School of Agronomy at the Federal University of Goiás, located in the Midwest of Brazil. The research was developed by an interdisciplinary group composed of 2 researchers from agrarian law, a researcher from geography, and myself, who researched in the area of nutrition and health. It is assumed that intensive use in Brazilian agriculture as a result of the Green Revolution is associated with numerous environmental and human health problems, including the increase in cancer cases. Thus, with the use of indirect data from government sources, we seek to relate the use of pesticides in Brazil with the increase in stomach cancer cases. To this end, through statistical analysis in an ecological study, data from the Brazilian Institute of Geography and Statistics, the use of pesticides by area harvest of soybean, corn, and sugarcane between 1979 and 2015 are correlated with the data National Cancer Institute of the annual incidence of stomach cancer mortality. It should be noted that the data chosen was the one available in the 2 databases. Still, the selected crops were those that, in addition to having available data, corresponding to 17% of the planted area of the country. Using the SPSS, the correlation and regression between the variables were performed, implying that the use of pesticides per harvest area of soybean, corn, and sugarcane, combined with the data on death from stomach cancer, revealed that the variables have a strong positive correlation, and the use of pesticides was a probable factor that contributes to the increase in the number of stomach cancer in Brazil during the last decades, configuring yet another evidence that the super-dependent chemical production model needs to be rethought. Despite the correlation, it is known that the other variables influence the incidence of cancer. Therefore, other studies should be analyzed together with this one. Thus, the research indicated a need to rethink public policies for the countryside, which do not encourage the use of pesticides, enabling the construction of an agenda that promotes food security and consequently better living conditions for Brazilians. Finally, thank you for time spent. I make myself available for qualifications. Bye.
Thank you very much, Nara. Let me ask you, so far as I know, the registration or legislation process in Brazil includes the tests for side effects on human health as well. So what do you think, how is this working, that still these enhancement of cancer increases?
Hi, good morning. This problem begins the us, and they are care more about the production than the people. I can see that, and we can think about it because we cannot preoccupate with them because we have a big problem, and the most people has susceptible to such comorbidity are the rural people who apply the pesticides, and they do not think about it. So I think they can are more rigorous than they can be.
You are now presenting this data on the Tropentag, which is far away from Brazil today, a little bit nearer than in other times. But how will you spread your results in Brazil?
We can publish it, but we do not do it yet.
Thank you very much, Nara. We will step forward, and I think we remain in Brazil. Manuel.
Good morning, my name is Rebecca. I am Brazilian and I study at Goiás State University. The present research project pesticide residues in Brazilian tomato, food security and President Bolsonaro deals with the exacerbated release of pesticides in Brazil. Its objective is an analysis of tomatoes, one of the most important vegetables in Brazilian daily consumption, and identification of legislative changes with greater repercussion in the rural belt. It also sought to detect the amount of pesticides approved since President Bolsonaro took office and to analyze the diseases caused by the consumption of pesticides. The study considered data on tomato consumption legislation projects and analysis in the Chamber of Deputies, data identified by Anvisa, the Brazilian Drug Regulatory Agency, and science articles. The research data evidence that tomato is among the leaders of overuse of pesticides in Brazil, having the second largest world position in cultivated area and the first in volume of industrialization. Sample of Brazilian tomatoes contained residues of HC8 and unauthorized organochlorine in Brazil. Law Project number 6299/2002 seeks to change the pesticide inspection body currently composed of Anvisa, IBAMA, and the Ministry of Agriculture. But with the approved law, it would become inspected only by the National Technical Committee on Phytosanitary Science Phyto. The project also proposes changes to several serious matters, such as production, import, and labeling. The proposal aims to reduce information available to consumers and to make it easier and to approve and import new pesticides. 624 new pesticides were approved since Jair Bolsonaro took office. Almost 30% of the ingredients contained in the pesticides released in 2020 in Brazil are banned in Europe. Consequently, there will be negative economic consequences for Brazilian producers. In a study carried out by Instituto Butantan at the request of the Ministry of Health, where Monica Lopes Ferreira was coordinator, it was proven that there is no safe dose of pesticides, even if proven regarding. The studies do not generate impacts of change in position in the legislative, especially because President Bolsonaro tends to discredit science and all sorts of data that could cause damage to his political allies or contradict his personal beliefs. The World Health Organization has registered 20,000 deaths per year due to the consumption of pesticides. Brazil is the country with the biggest Conception of Pesticide Science 2008. The acephate used in table tomatoes has already been proven to be related to diseases such as leukemias, non-Hodgkin's lymphomas, and pancreas.
Thank you very much, Rebecca. This was more a political statement than a scientific one, and I don't want to discuss the political side. But every time the production is related with exportation, with selling the goods, so let me ask you concretely, what does this overuse of pesticides mean for the exporters in Brazil? Tomato is exported to Europe, I think, as well, and they would not like to let the products enter with too high residues. Couldn't you hear me? You have to unmute your mic. There seem to be some problems, so we are coming back later to you. Maybe the Brazilians can remain in the Zoom session after we are no more online, and we could discuss this in more detail. Manuel, your turn for the next presentation, please.
Hello, my name is Tanya Raynand. Welcome to my presentation on bacillus-mediated changes in iron partitioning in lowland rice under iron toxic conditions. The aim of the study was to find out whether changes in iron partitioning could be the reason for the effects of bacillus inoculation on leaf fronting scores. In the first step, we screened different cultivars for their response to bacillus inoculation. As seen in this graph, the effects of inoculation on leaf scores were quite contrasting for different cultivars. For Swakuku 8, a moderately tolerant cultivar, inoculation leads to higher scores, while in IR31, a sensitive cultivar, the inoculation leads to much better scores. So the question was, could these effects be due to altered iron translocation into the shoot? Other than expected, the share of shoot iron was not decreased but even slightly increased in IR31 despite the much better leaf scores. So a look at the next graph shows that shoot iron concentration is indeed not correlated with leaf fronting. So we had a closer look at the leaves themselves. Is there maybe altered iron partitioning into the leaf blade and the sheath? The upper parts of the bars in the third graph represent the iron content in the blades of the three youngest fully expanded leaves, while the lower parts represent the sheath. The numbers within the bars give the share of total shoot iron content. We found that the ratio of iron in the blade to iron in the sheath is always lower in inoculated plants as compared to the controls. So there is more iron stored in the sheath, but however, this is the case for both cultivars, and mind you, inoculation had contrasting effects on the scores. So we had an even closer look at the iron in the blade. And although we see less iron in the Cumulus inoculated IA31. We could not find a general correlation between staining intensity and overall plant score or symptoms on the harvested leaf area. So in conclusion, we found that Bacillus Cumulus and Bacillus megatherium isolates can have contrasting effects on the tolerance to iron toxicity in different lowland rice cultivars. The shoot iron concentration does not always correlate with leaf Brunson score. Iron storage in sheath can improve tolerance to iron toxicity. The iron content in the leaf blade, however, does not always correlate with leaf symptoms. So it's quite obvious that there are other mechanisms involved as well. So the impact of bacillus inoculation on Ross scavenger enzyme activity will now be investigated. Thank you very much for your attention, and I'm looking forward to receiving questions and comments. Thank you.
Thank you very much. So, what can we learn from it? I didn't understand correctly. Where did you inoculate the bacillus? To the soil or to the leaves? There are some problems with Tanya, but
How?
so you can write it down.
I tried to use it.
That means in a hydroponic system, you have bacillus in the soil and in a nutrient solution, and that can mean that you are observing a plant growth promoting bacterium, and this PGPR may be a product afterwards, could be produced and then separately inoculated in the field as well. We know this from Brazil, no? I learned that the stream cannot read the chat, therefore I repeat the answer of Tanya. She said, yes, it is possible that bacillus can be produced afterwards routinely and could be inoculated to the field environment as well as a product, for example, to enhance the iron uptake of rice. Are there questions from the auditory? Only where we can get the presentations. So, okay, the auditory may participate in asking us via the session Q&A. There is one question, what is multifunctionality? But we still not discussed this point here. Maybe bacillus is a good example to discuss multifunctionality, because bacillus can be used as a plant growth promoting bacterium, but at the same time can directly interact with antagonists and can be an antagonist for pathogens. Did you think about this aspect too, Tanya? She says, for now we are using it to study tolerance mechanisms of rice against iron toxicity. It is about reducing uptake. Our results do not show a PGP, but we screen resistance against fungal disease. Yeah. And this would be an example for multifunctionality. You are using an organism not only for one aspect, but you are trying to find out which effects does he still have against other things, for other things. And so you would describe the ecological niche of this organism and create the possibility to use multifunctionality of this organism if you are using it as a product. Thank you very much. Tanya, so we have still someone on the list. I would like to ask Manuel if we have one more presentation. Dr. Jacobo, I think, did not appear. Is that right? No, it was the last, it was said. So let me ask you all, what might be the reason that the discussion of plant protection on the Tropentag is decreasing every year? Are there no more real problems in plant protection all over the world? You specialists. You auditory, why are you listening us? Why not all the 700 people are listening us? And what can we do better next time to attract more people for plant protection? Is plant protection not important? Minimally at the Tropentag. I would like to learn it from you. To say this, I'm the managing director of the German Phytomedical Society. That means the Scientific Society for Plant Protection and Plant Health in Germany. We have a working group on tropical and subtropical plant protection since 30 years, since 1994. And the interest in Germany in the tropics is not decreasing, but it is a matter of a distribution of interest. There are no longer centers which are really working with plant protection in the tropics. You, Tanya, from Hohenheim, know that you are only laterally working with plant protection, only working on plant nutrition and so on. But we lose, therefore, the contact with all the single initiatives which are underway, and we do not know who is working where about what. And if we are not going to the Tropentag and we are not coming together at the Tropentag, presenting plant protection aspects as well, we as Germany will lose the knowledge where to interact on the area of plant protection. This might be no problem because each single country can take over the own problems, but this will influence the politics, helping only on the economic action area, for example, or interacting in other ways, building streets and not longer taking care for food. Okay. Itorobong tells us plant protection is very important as there is increased reliance on chemical pesticides, which in the long run has damaging effects on the plant consumers. That's very true. I am astonished that you write increased reliance on chemical pesticides. We, in this comment, we can see that the world maybe is divided in two halves. In the north part of the world, the reliance in chemicals is decreasing. We are looking for possibilities to avoid chemical pesticides, find a way for agrobusiness which is not relying on pesticides. We interpret the IPM legally based. We have a directive in the European Union which defines the IPM as something what is composed of a huge amount, minimally 10 different components and elements, and we try to agree about the sequence which has to be followed until at the final stage pesticides can be used. And this is a absolutely different way of thinking, which we try to establish since 30 years, since 1986, I would say. But if you are writing now, Itorobong, that the reliance on chemical pesticides is increasing in your countries, you should see that you especially have the chance to avoid all our mistakes now. We had a huge amount of mistakes made because of relying on the pesticides. The examples we saw here in the session, using multifunctionality of microbes in the soil, which are increasing the nutritional situation of plants, but at the same time may influence the resistance reactions of the plants as well, or act directly as antagonists for soil microbes which are detrimental. Or the example of Brazil, which was not presented, which was shown here or applicated first by Adriano Nascente, that Brazil is using a huge amount of plant growth promoting bacteria already in agriculture. I'm working together with someone from Passo Fundo and can see that it is really very interesting to see these effects, which are reducing the amount of pesticides if they are used consequently. Furthermore, the selection of cultivars we had. Yesterday we had in the session, in the oral contributions, examples how to select cultivars which are resistant or more tolerant to stresses, biotic and abiotic. These are elements of IPM, and these IPM is possible without costs in African countries, in Brazil and wherever, all over the world. These basic elements of IPM have only to be followed consequently in a certain row. Start with the selection of cultivars and then continue with biocontrol, with phytosanitation, with phytocontrol, with biocontrol, with vegetation control, with push and pull techniques, and so on. And finally, if nothing can be solved with this simple techniques which are often relatively cheap, then you can use pesticides, but only pesticides which are legislated, which are tested not to be harmful to health, which are not harmful to environment, and so on. And this, of course, the background of the single country you are living in has to be, or has to reflect and has to ensure a certain legislation process has to be at the beginning of the approval and the risk assessment of such pesticides. And then everything can be good. We in Europe see that even then it may be very bad because of the misuse of pesticides which takes place on the level of the farmer. But if the society is educated enough and understands, is aware of all the problems which pesticides can create, then a balance between the positive effects of pesticides and the negative effects takes place, and we can use them without too much risk. So I have some comments here. One moment. Although in the Western world there is more awareness on organic products, but in our countries this awareness is still very minimal, and we are most concerned with agricultural output. That's right, Itorobong, but the output is not necessarily lower in organic agriculture than under the use of pesticides. Of course, we have to find the ways how to produce as sufficient as we need. But I'm personally working with permaculture, for example. Permaculture in cities, that's my profession. And we could learn by using the correct amount of compost, recycled biological material, to increase the production over 300% in comparison to not using this compost without any agrochemicals. So I'm very convinced that this is a very interesting approach. And we try this in Sierra Leone as well, introducing permaculture in Sierra Leone. So I think we lost a lot of experience. We lost a lot of knowledge. Maybe old people still have the old knowledge. We younger, and in that moment, I beg your pardon, we lost a lot of knowledge, and we have to recreate and rethink everything. Maybe every 30 years we have to do it in the same way. But hopefully we are respecting the old ideas. We are not losing everything and can step forward slowly and come to to a solution which optimizes the situation in which we are. So I would say we do not have more questions. Oh, and Tanya, thank you very much, Tanya. Tanya mentioned the Moringa project. Yes, she saw the presentation, I think. Yeah, the Moringa project, to tell you this in a short moment, is this permacultural project in Sierra Leone. There we try on a degraded site to establish an agroforestry system, and we try to establish what we call a citizen science approach. Maybe this is a very important point to make here as well. Citizen science approach means that the farmer himself is collecting data, for example, by smartphones. We learned it from other posters, from other oral contributions, that this might be very easy to establish. And after collection of the information, together with scientists, he is deciding what to do next. He learns more about his own system because he can document what he is doing and he is monitoring what is happening. And afterwards, hopefully, is deciding on basis of these solutions. And doing so, he is able to inform other farmers about his experiences. And someone else, I think it was Lawrence Bachmann on this Tropentag, he said this would be a farmer trainer. A trainer who is a farmer and trains other farmers. And these farmer trainers seem to be very effective and can create situations which are not only based on the economic outcome of a farming system, but as well highlight all the other aspects like health and so on. We have to learn to produce healthy food, but in a sufficient amount, as you said, Itorobong. No? And we should not rely too much on pesticides and rapid growing cultivars, which are offered by multinational companies as well, and increase the amount of seeds which we are collecting, and should base our production on land races we have adapted to our environment and so on. So more independent growth of plants, more independent growing systems which are locally adapted are hopefully a better solution than a generality of design of plant production systems. For example, I sampled the number of cultivated plants on the markets offered in 1975, 1995, and 2015 here in Germany. And we saw a decrease of numbers of different plant species. Why? Because not only all over Germany, the same carrots were planted and the same tomatoes, but this disease was spread all over Europe. We went to the south of France and found the same varieties like in north of Germany. That's incredibly and that's stupid. So to adapt the production systems to local conditions is a much better solution. It's not necessary to avoid every high production cultivar, of course not. But you have to find your own way for your own regional aspects. And you should speak with the farmers from the surroundings, which are supporting the urban areas, for example, as well. So find a solution which is oriented in the own demands and not every time looking to export possibilities and so on, feeding the north, feeding the rich countries. Regionality first and then the overproduction for the export. And I can't understand why most of the world is accepting different residual levels and is not arguing that there should be the same maximum residual levels for all. Look to the mycotoxins. Africans have to accept a higher residue level than Europeans. Why? Why is it like this? Yeah. So, we have only some minutes left. Are there further questions? This is not the case. Tabea.
It goes into a bit of another direction, but it would be a question to the Brazilians, since there's been multiple inputs on the pesticides. And I've just been wondering if there are any concrete suggestions of what could be done on the policy level to improve the situation at the moment. So in a way, I think from an ecological perspective, we're often thinking of zero pesticide would be the best. But then when we think about an social perspective or economic perspective, sometimes that's not possible for the farmers, since they would lose all their crop and would not have an income anymore. So I'm wondering, do you have any ideas of what could be done on the policy level to improve the ecological situation without hampering the social and economic situation completely in a sense?
And we think if the police had more incentive for this, we can amplify the culture of agronomy and simplify the process.
Yeah, thank you very much. I think incentives is probably a good approach to start.
Yes, yesterday we had an interesting talk, and someone was showing a formula and showed, in this case, someone will change his behavior. In this case, he will not change the behavior. And the decisive point was money. That was really interesting to see. And of course, it is correct. Money in biology is energy, and you can change the word money with a lot of advantages, in contrast to the former case, and that's really important. We have, in any case, to find something what is attractive for those people we want to change in their mind or in their behavior. Okay, thank you very much, all of you. I invite you, Six, to remain a little bit. We are now, Manuel, telling the audience that this session will close. And we thank you for your participation. We didn't hear so much of you audience. Please participate in further sessions a little bit more. And nevertheless, thank you very much for participating at Tropentag in this session. Goodbye.